Hoje, no Entrevistando Cientistas, vamos conversar com a doutora Vousden, graduada em Genética e Microbiologia pela Queen Mary University of London, onde também concluiu seu doutorado em Genética. Atualmente, é cientista-chefe no Cancer Research UK e líder de grupo no Instituto Francis Crick, no Reino Unido. Sua pesquisa é focada principalmente nos mecanismos moleculares do câncer, com ênfase na proteína P53 e no papel do metabolismo celular na progressão tumoral.
Você poderia nos contar um pouco sobre sua trajetória científica e como começou o seu interesse pela pesquisa em câncer, especialmente pela proteína P53?
I actually started out doing a PhD in fungal genetics, but then just accidentally, really, for my first postdoc, I moved into cancer biology. First, when I started my first postdoc, I was working on identifying oncogenic versions of viral human oncogenes that were the homologs that were found initially in viral proteins, and that's through that we identified the RAS oncogene. And then in my second postdoc, because I was used to looking for transforming oncogenes in different situations, I started to look for oncogenes in human papillomaviruses that were associated with cervical cancer. Because at that time, it wasn't really clear whether it was the papillomaviruses that were really causing cervical cancer or whether it was herpes virus. So during that work, I identified two of the viral oncoproteins, E6 and E7. And just at the time that I was leaving that postdoc, Peter Howley's lab was showing that the E6 viral protein functioned by binding and inactivating P53. So we got really super excited by that. And it was just at the time when P53 was being described as the tumor suppressor, and it became evident that P53 was mutated in many different human cancers. So we started to work on how P53 functions as a tumor suppressor. We were able to help to show that P53 is regulated by MDM2, which targets it for degradation. And we also helped to understand exactly what it is that P53 does. Not that we know exactly what P53 does now, but we contributed to, lots of large body of data generated by many people describing what P53 does.
No seu trabalho de 2001 com Katsunori e Nakano, vocês identificaram a puma como um novo gene pró-apoptótico induzido por P53. O que os motivou nessa busca? E na época, chegaram a considerar outros genes com potencial semelhante ao da puma?
Yes, so we actually identified puma. Cats described puma when we were all working in America at the NCI, and we knew at the time that P53 was a transcription factor, and so we figured that probably P53's activity would be explained by identifying genes that would be induced by P53. So Katz carried out an expression array. It was back in the old days. We didn't do RNA-seq in those days. And he found, obviously, a huge number of things that were inducibly expressed after he activated P53. And he spent a long time validating all of his data. And in the end, he found 3 genes that he was very confident were genuine targets of P53. So one of them was actually a subunit of ribonucleotide reductase, which probably contributes to the ability of P53 to help during DNA repair. But just as we were working on this, the paper describing this as a P53 target gene was published. So we were scooped on that. We did publish it eventually, but in a much smaller journal. The second gene that we found was puma. And immediately we could see, of course, that it was a PH3 domain protein. So it was pretty obvious what it was doing. And so we worked that up. And during the course of doing that work, we found out that Bert Vogelstein's lab had also identified the same gene as a P53 target gene, but they hadn't published it, luckily. So we contacted Bert and call it puma. And so we published coincidentally with Bert's paper. And I think, it just became a BH3 domain protein that was clearly important in mediating this apoptotic activity of P53. I'll tell you, the third gene was incredibly confusing to us because when we expressed it in cells, it actually seemed to help cells survive apoptosis. And of course, at the time, we were just very convinced that everything that P53 induced would need to be pro-apoptotic. And so we stopped working on it for a long time because we couldn't understand it. But then many years later, when we were in Glasgow, we started to understand that that gene is actually part of an antioxidant defense that helps cells avoid apoptosis. And we called that Tiger because we had Puma and now we have Tiger. Played into this growing realization that p53 is not always killing cells, but can actually also contribute to survival functions to help cells survive under different types of stress.
A via regulatória P53-MDM2 tem sido amplamente estudada com várias tentativas de direcioná-la terapeuticamente. Na sua opinião, essa via ainda representa um alvo viável para o tratamento de câncer? E caso sim, quais são os principais desafios?
Yeah, so MDM2 targets p53 degradation, and very quickly after that, some of these MDM2 inhibitors started to be produced. And it's actually a really interesting example of how you can make a small molecule inhibitor that actually inhibits protein-protein interaction and is very effective and efficient and specific. And that's because of the structure of MDM2 and the way that, you can dock a small molecule. So very quickly, we had drugs that worked incredibly well, and we were all super excited. And so the fact that they're not really yet part of standard cancer therapy is very disappointing. And, there's been various reasons why that might be. Some of them are related to on-target toxicities that, just reflect the fact that if you activate p53 in normal, sensitive, radiosensitive tissue, you'll induce toxicities. And another problem, I think, is that the approach obviously depends on there being wild-type p53 in your tumor. And we know that it's quite easy to avoid the activity of wild-type p53 simply by mutating p53. So there's a very clear, effective resistance pathway that tumors can use to overcome their sensitivity to these kinds of therapies. So I think I think that there still is hope, but I think it's been difficult. It's been a difficult path. But I guess on the other hand, given that so many cancers have mutations in p53 and they're generally not found in normal tissues, I think that sort of provides a sort of tantalizing possibility that you could target mutant p53s. And there's been a lot of discussion about whether mutant p53s have any gain of function that would promote tumorigenesis, and that maybe would it be a good idea to try and inhibit those activities of mutant p53. But I think maybe the most elegant approach is one that a few people have been taken to try to find small molecules that actually bind to mutant p53 and refold it so that it regains some wild type activity. And that's really appealing because it takes advantage of the fact that mutant p53s are generally only in cancers. There is one such small molecule that's in clinical trials at the moment. The slight problem there is that it only refolds one particular p53 mutant that's not very common in cancer. So, it would only be applicable to patients that had tumors with that particular mutant. But I do remain optimistic, I think, that if we can understand p53 a bit better, then that will lead to the development of some exciting and maybe effective new therapies.
Sabemos que o microambiente tumoral influencia a disponibilidade de nutrientes e que as células cancerígenas apresentam grande flexibilidade metabólica. Na sua visão, de que forma esses fatores podem representar desafios para a eficácia das terapias baseadas na restrição de aminoácidos?
Right. So we've been very interested in this, and as you say, we've spent many years looking at the tumor cells themselves, but it's really clear that there are some very interesting interactions between cancer cells and all those normal cells that surround them. And in multiple cases, we can see now that the normal cells around cancers can be co-opted by the tumor and making them produce nutrients to feed those tumor cells. We also see that cancer cells can deplete nutrients and so, maybe hamper the normal function of things like the immune cells that would otherwise be able to attack and clear the cancer. So I think, obviously, in terms of nutrient acquisition, we need to fully understand how different cancers acquire nutrients and how that relates to the type of cancer, where the cancer is, the genetics of the cancer, and possibly even the type of therapy that's being used. So probably, as you know, we've been very interested in the amino acid serine. So we know that cancers need a lot of serine, but we also know that there's many different ways in which cancers can satisfy that requirement. So some cancers can upregulate their intrinsic serine synthesis pathway. So that means that they can make their own serine and they don't particularly care about exogenous serine. So in those tumors, obviously restricting serine to them may not be very effective, but they could be incredibly responsive to small molecule inhibitors of the serine synthesis pathway. So that's kind of one group. For some reason, there are another quite large group of tumours that have lost the ability to make their own serine. And I assume there's a selective advantage to that, but I don't think we really know what that is. But they are now very dependent on exogenous serine. That exogenous serine could be provided in the circulation, could be in the tumour microenvironment, in which case dietary depletion of serine could be effective. And there are clinical trials ongoing that are using serine-depleted diets, but only in patients that have tumours that are not able to synthesize their own serine. So there's a selection for tumours that might be very sensitive to exogenous serine. But then, as you so rightly say, stromal cells in the tumour microenvironment can also provide serine to cancer cells. There's a paper showing that neurons can provide serine, and we have some data as yet unpublished showing that adipocytes and adipose tissue can provide serine. So I think we need to know a lot more about when and where this happens and which stromal cells are responsible. And once we know all of that, then we can start to think about how we might be able to intervene in this for therapy. But personally, I'm quite enthusiastic about nutrient limitation in cancer therapy.
Como estudantes iniciando nossa trajetória na pesquisa biomédica, qual conselho você daria a nós, jovens cientistas?
You have to love what you do. You should always follow your heart. So if you're passionate about your work, if you're excited about it, if you want to go in the morning, if you feel real pleasure from, that getting a good result, then that's the right job for you. Don't worry about the future too much. Don't worry about what's your pension going to be. Don't worry about these things. Worry about enjoying the work that you're doing. Obviously, as you know, I'm sure you're all well aware, things don't always work, experiments don't work, and that's fine, and that happens to everybody, and there will be times when you feel depressed and unhappy, but fundamentally, if you're excited by it and you love it, and honestly, it is the best job in the world, and then I think you just enjoy.